So good afternoon, everybody. My name is Stephen Burley. I'm the director of the Institute for Quantitative Biomedicine and the interim director of the RAD Collaboratory. And I'm very pleased to welcome all those in the room and those online elsewhere in the United States and in other parts of the world to today's IQB seminar. We're very fortunate to be hosting Dr. Josefina del Mármol, and I'll say a little bit more about her in a moment. If you could move to the next slide, please. We'll just complete some housekeeping announcements. So we are second to last for our full series. On December the 10th, Jay Srebrenik has kindly agreed to step in, having lost our NIH speaker to jury duty, of all things. Not some NIH ruling, but he's been called for jury duty. So we are going to be graced with Adrian Ferry-Demare's presence in the fall of 2026, I think, because the spring's already full. So next slide, please. I wanted to draw to your attention an opportunity to participate in an AI for Data Science at Rutgers Day on December the 8th in Newark. It'll be at an event space known as Express Newark, which is on the Rutgers Newark campus on Halsey Street. Interesting art and maker space, I'm told. The RAD Collaboratory will be well represented there. The symposium is going to bring together the Rutgers leadership, faculty, students, postdocs, and partners from industry, the community, to talk about the Rutgers Roadmaps initiative, of which the RAD Collaboratory is one. And a couple of people in the audience will be helping with that, and I am very grateful for your willingness to do so. Next slide, please. I also want to draw your attention to the fact that RAISE 26 will take place this year. And Professor Jim Samuel in the Blaustein School of Planning and Public Policy is running this opportunity to win over $20,000 in prizes. The registration deadline is the 8th of December, and you can see the URL on this slide, and there's also a QR code being displayed if you're interested in participating in this AI informatics data science competition. Next slide, please. The RCSB Protein Data Bank will be holding another virtual office hour on the 24th of November. That's the Monday of the Thanksgiving week. Our Joan Segura will be presenting on exploring computed structure models at rcsb.org. For those of you who don't know, if you go to the top right-hand corner of the rcsb.org website, there's a little toggle that allows you to get access to more than 1 million computed structure models in addition to the nearly 250,000 experimentally determined structures that are currently housed in the PDB archive. Working with both computed structure models and experimental structures can be very informative, and I urge you to explore that opportunity and attend the virtual office hour. Next slide, please. So this brings me to introducing Josefina. She received undergraduate training in biology at the University of Buenos Aires. She's a native of Argentina. And we worked out that our paths may actually have crossed in the Los Alerces National Forest many, many years ago. She was there camping and enjoying the lake and the scenery, and I was there fly fishing. An amazing part of the world. She was then had got the opportunity to join the lab of Nobel Prize winner Rod McKinnon at the Rockefeller University for graduate study, received a PhD in molecular neurobiology and biophysics, and she was supported by pre-doctoral fellowship from HHMI at that point, and she was working on the molecular basis of the sense of touch. So those are proprioceptive channels in the trade. a short postdoctoral stay at UC Berkeley, then saw her moving back to Rockefeller to join the lab of Vanessa Ruta to study the structural basis of odor recognition. That's what we hear more about today. In 2022, Josefina joined the faculty at Harvard Medical School as a tenure-track assistant professor. She is the recipient of a number of awards and funding, and I'll just briefly run through this impressive list. So she has a NIH Mosaic K99 Pathway to Independence Award, the ACHEM's Polak Young Investigator Award, Tri-I Breakthrough Prize for Junior Investigators, most recently having also received the Blavatnik Regional Award, and is again supported by the Howard Hughes Medical Institute as a Freeman Harabowski Scholar. So it's terrific to have somebody who's already so accomplished at such an early stage in the career. We'll hear today about structural mechanisms of odor sensing in insects. Now, what this really boils down to is how does the mosquito smell you? And that's what we're going to hear. So welcome, Josefina. Great to have you here.
Thank you so much. Okay. Thank you so much for having me. Thank you so much for inviting me to come here. It's my first time at Rutgers, and I've, of course, known about it for so long. I've been in the area or I was in the area during a lot of my training. So it's a real honor to be here. As Dr. Burley mentioned, I've been interested in sensory processes for most of my career at the molecular level. And today I'm going to focus on olfaction, and I'm going to try to convince you. So I'm going to try to tell you why olfaction to me is so interesting. What I've done here, what I put in the slide is I grabbed all of the sensory receptors in the human genome. So these are all the membrane proteins that are in the membranes of the sensory neurons that transduce some sort of stimuli. And so you can see we've got about 8 opsins that are responsible for vision. The 2 piezo's, this is an unrooted alignment, by the way, so it's somewhat to have an idea of the relationships. These are the piezo's responsible for most of our touch, the TMCs, responsible for hearing, some 15 of the trip channels, of the more or less 30 trip channels that are directly responsible for sensing things like temperature and osmolarity. And then we've got about 500 chemosensory receptors, and this encompasses olfaction and taste. And so what is very clear is that there is something different about the nature of olfaction and how we have to deal with this sensory information. And this really highlights a fundamental difference in these senses that comes down to the physical nature of the stimuli. Because at the very basis, transducing things for vision or hearing is essentially dealing with a wave, either a sound wave or an electromagnetic wave. Yeah. Yes. Okay.
I can fix that.
I can. Yes. I
can off it.
Okay. I can project. I can try to be more a little louder. But let me know if you can hear me. Okay. All right. So what I was trying to get at is that the physical nature of the stimuli that gives us vision or hearing is a wave. And a wave has a wonderful quantitative, sort of continuous features, parameters that you can use, you can rely on, and have a direct representation in a brain. For example, wavelength, this just one single parameter is directly related to how we perceive the note or the color in the stimuli. Now, there's nothing as mathematically simple to describe the sense of smell, because odors fundamentally are a collection of millions, possibly billions, of volatile molecules that are entirely distinct from each other. They have no parameter that is continuous that connects them, and there's no relation between the structure, the formula of an odor molecule and what it smells like. For example, this is what these molecules smell like. That really has we have no ability to predict what this is. And even if we could, they don't behave in any predictable linear way. We don't really have an equation to deal with this information. So all of these molecules together, for example, some of them are pretty unpleasant, classic putrid smell, sulfurs. We put them all together, and what you get is the sense of the smell of coffee. So obviously there is something really challenging about the sense of smell, something that animals have to cope and have to find a way to encode so that we can actually make sense of this information and use it, because it's incredibly important to navigate the environment. Sound good? All right, great. Okay, so here's a challenge. How do animals encode chemical information? Extremely diverse, with an enormous amount of physical and chemical variability. So what do we know, or what did we know when I started this work? Some general guidelines are common across animals, and I'm going to tell you a little bit of what is really seems to have been a general way to encode chemical information. Most animals have some set of receptors. In our case, it's about 400. Typically, it's a pretty large number, and they detect a subset of the chemical space in what is sometimes referred to as a combinatorial code, where most receptors can detect multiple molecules. Some are very specific, but they have overlapping sensitivities, so that the identity of any one odoram molecule is encoded in the pattern of activation of the odoram receptors that interact with that molecule. So we have some receptors that you can describe as sometimes they're called as very narrow specificity, some that are very broadly tuned. They can interact with a lot of molecules and have sort of different tuning curves. But when I started working on olfaction, the main challenge or the main difficulty or really question is how these differences arise at the protein level. What are the structural principles that give rise to the very different tuning curves of odoram receptors? And the reason why this was elusive at the time is because we had not, as a field, been able to study these molecules at the atomic level. They're very hard to work with. They don't crystallize. They're very, we don't make enough to be able to even try to crystallize them. They're very finicky to work with, and it's just not biochemically tractable. So essentially, we had no structures. We had no really atomic view of odoram recognition by odoram receptors. So I started working on this at the end of my postdoc and now into my lab. I focus on insect olfaction for a variety of reasons. Some of them are very visual, and I love these pictures because it really highlights that some of the most impressive structures in the insect anatomy are the antennae, which are these sometimes plumose structures on the front of their heads. And they, just by the shape, you can tell how important this sense is and how incredibly diverse their olfactory mechanisms are. So insects give us a lot of good proteins, interesting proteins to work on and to understand olfaction. The other reason is because, as Dr. Burley mentioned, insects use their sense of smell to find their host. For example, mosquitoes use the sense of smell to hunt us, to bite us, and this is how they transmit a host of deadly diseases that cause really havoc in an enormous part of the world, including where I come from. So understanding how mosquitoes smell us, for example, would perhaps help us curb the spread of these diseases by preventing bites and also make picnics a lot more pleasurable. So maybe some of you might be familiar with mammalian odoram receptors. These are classic GPCR proteins. They are membrane proteins that upon binding an odorant, they will initiate signaling through classic GF-OFF pathways. Now, in vertebrate olfaction, it has known for about 20 years now that it relies on a completely different mechanism. Unfortunately, insect olfactory receptors also have 7 transmembrane domains, which made it very confusing because for a long time, people thought that they were also GPCRs because it would have made sense, and they're not. And it took quite a while for the field to come to terms with this. These are ion channels. Upon binding an odorant, they initiate signaling through passing ions, depolarizing the neuron, and calcium signaling. So when I started my work, this is pretty much all we knew about it. We had no structural information on any of these. And I really wanted to look at the binding pockets. I really wanted to understand how do 400 odoram receptors in the human, for example, detect billions of odoram molecules? How is that happen at the chemical level? So I took, in order to sort of circumvent some of the challenges, I took an approach that I learned from Rod, and I think it's a very interesting approach, which is sort of an evolutionary road trying to find proteins in the animal tree that would be more amenable, more tractable biochemically. So instead of really trying to crystallize the most interesting odoram receptor in a mosquito from the get-go, you just look in the tree for something that perhaps behaves a little better, and you can start understanding the family of proteins. So doing a very large screen on evolutionary related proteins, I went all the way back to the insect tree, and landed on this species, is the bristletail, that is really, I think I have in the slide, it says ancestral. I hope there's nobody from the evolutionary field here because they're not ancestral. All of these are modern proteins in that they all exist, but they're earlier derived, so they're more at the base of the tree. So oftentimes they're just referred to as ancestral. So the bristletail odoram receptors, bristletails only have 5, and they were biochemically very tractable, meaning they expressed really well. I'm not going to show you all the screen because it'd be incredibly boring, but they just have an incredibly solid, tractable behavior on the bench. So that was fabulous. First problem is, what does this animal smell? Who knows, right? This is not a model organism. So the olfactory life of a bristle tail was not really something that it had been studied. So what I had to do is I had to take these 5 odoron receptors and find out what they bind to, what they smell. And so I developed some assays. Again, these are ion channels. So I use calcium signaling and electrophysiology. And I was able to deorphanize 2 of these odoron receptors. And I'm going to focus on only one of these because it's the bulk of the work on this part. And so this odoron receptor OR5 from the bristle tail, it detects a variety of different odorants. So it has a response to a panel of odorants. The largest response we got with eugenol, which is the smell of cloves. But it also smells a bunch of other things, such as lemon, some citrusy smells, and dirt. So geosmin, the terpene on the end, it's the smell of rain, sometimes it's called. It's kind of a musky smell. So this was, for me, a really exciting opportunity to look at a binding pocket of an odoron receptor that is able to interact with anything from aliphatic compounds to aromatics to terpenes with various degrees of sort of chemical functional groups, and understand how that happens. What is going on in the binding pocket to be able to give you this response? The other reason this was interesting is because it also detects DEET. And for those of you who might not know, maybe many of you, DEET is the active ingredient in OFF, the insect repellents that we all use. And I don't know if this is going to blow your mind, or maybe you already know. We actually have no idea how OFF works. I don't know if you know that. When you use it, it obviously works. Mosquitoes don't like it. No idea why. We don't know what it binds. It wasn't even clear if it was olfactory or not. It's been around since 1940. It works. We don't know why. So this is actually, it was suggested that it was an olfactory pathway, and it was pretty clear that the volatile phase of DEET is repellent. So this is the first possibility at really getting at the interaction between DEET and an odoron receptor and really showing whether it actually binds odoron receptors and it activates them or it doesn't. So this was a great candidate for me. So I was able to obtain quite a bit of this protein, and thankfully this was by the time, so this was 4 years ago, cryo-EM had already bloomed, so we were able to, with a fairly small amount of protein, get a really beautiful structure. So this is, I'm just showing the grids because anybody who's in the structural field loves to look at the actual images. And so this is how the protein looks like. It's a membrane protein. So if you look at the particles, mostly you see the micelle. It's very much embedded in the membrane. There is very little outside, so you can, that sort of UFO shape is the detergent micelle, how I purified it. And so doing the classical pipeline of cryo-EM, we got really beautiful 2D classes right away. You can see even sort of the TMs through some of these classes. And then this gave us a beautiful structure. For me, it was the first structure of an odoron receptor and the first one that I had seen. We know when you get your structure, if you're working on this for a long time and you actually see your density, and it's really, it's quite a moment. And I'm just here showing that you could actually, the resolution, which was sort of overall between 3.1 and 3.6, so now nominally 3.3 angstroms. So in this case, it really allowed us to model the majority of the side chains with quite a bit of confidence. It was pretty good density. So this was the first structure of an odoron receptor. And this is what it looks like. This is a homotetrameric ion channel. It has 4 subunits. They're all the same. They're colored differently just to show you so that you can see the differences, but they're all 4 copies of the same subunit. And so really, when first we got this, we really wanted to know how it works. You can see the majority of it is embedded in the membrane, and a very small fragment is intracellular. If you turn it around and look at it from the extracellular space, you immediately see the ion conduction pathway, the entryway of the ion conduction pathway, because again, this is an ion channel, so it's very good that it has an ion conduction pathway. And then obviously the first thing I really wanted to do is to get structures in different states to understand what the binding pocket looks like and how it opens the channel. And so the first thing to do, this is the view from the extracellular side that I was showing you earlier. This is the apostate, the unbound. There's no odorant here. And if you focus on the entryway, the ion conduction pathway, it's fairly closed. If you solve the same structure in the presence of eugenol, which is the top ligand, immediately you can see a dilation of the ion conduction pathway, mainly at the top. And again, because this is an ion channel, if you record, you can see these beautiful currents. This is in a patch. You can start to see single channel recordings. And this is not a single channel patch. It has many of them, but you can, I just zoomed into some of these traces, mostly to show something very interesting about this channel, which is it's a very small opening. It's a very small conformational change. When you add eugenol, very few changes happen, and most of them in the pore. And this sort of matches the behavior of the channel, which is very flickery. It has a very, very fast opening and close. So it's a very small transition, and it's compatible with a very sort of almost like a breathing movement at the gate. And then if you solve the same structure or the same chlorine in complex with DEET, which is the other ligand that was very interesting to me, you see the same, so different ligands end up in the same overall structure, the open structure that has the same open diameter and overall the same conductance properties. And so having an apo, two bound structures, now somewhat validating structurally that we can, in fact, open the ion conduction pathway with the ligands, we went ahead, and this is just to highlight what I mean when I say the opening, because it's a very small change, so you can highlight there. So obviously the next thing to do, the next place to look at in the protein is the odor binding pocket. That's really what I wanted to look at. And I'm leaving aside a lot of the mechanistic work that we did on this protein, mostly to, well, I can't. Okay, I'll read that later because it's just you want to know the question. I'm not going to talk about a lot of the mechanistic because I'm going to focus on odor and specificity right now. Otherwise, we spend a lot of time talking about the rest of the protein. So in terms of odor and specificity, where is the odor binding pocket? So the odor binding pocket is if you take a slice through sort of about 40 angstroms through from the transmembrane domain, you see this. Can you see my cursor? I guess you can. So you see very clearly in the apo state this sort of pocket in the middle of the transmembrane domain. And if you look at the same spot in the presence of eugenol or DEET, you very clearly find the densities for eugenol or DEET. Here are the models, and I'll show you the densities in the next slide, which the first surprise was these are in the same binding pocket. A lot of people had predicted that the way by which another receptor can be broadly interacting with many molecules is by having different binding sites. So maybe all of them have different chemical specificities. In these and other structures and through a lot of mutational work, it's the same binding pocket that binds all of these molecules that I work with for these 35 ligands that activate this channel in my panel. All of them bind to the same binding pocket. So that's really interesting. And how does that happen? How can a binding pocket interact with so many different molecules? So here's the detail of sort of the atomic details of the binding pocket. And here I'm showing the densities because it's very clearly different density for eugenol and DEET, which was really challenging because it's a very small molecules, they're very hydrophobic, they're not very tethered inside the binding pocket. So it was required a lot of particles and a lot of data processing to really have the certainty on the densities that we were looking at. So here we're looking at the binding pockets in the presence of eugenol, in the presence of DEET. And what I hope you can appreciate is that the binding pocket is this beautiful, somewhat small, concise cavity lined with hydrophobic residues, a lot of aromatics that give us sort of sites and some sort of rigidity, and some aliphatic hydrophobic that confer some flexibility. So what was really interesting is that aside from some difference that I'll highlight, the binding pockets look very similar in the presence of both ligands. So how can the same binding pocket in a somewhat similar configuration give rise to different responses to eugenol and DEET? So if you look at the activation curves of eugenol and DEET, maybe I should have overimposed them to show you more clearly, but eugenol has much left-shifted EC50 and higher activation. So it's a better link, it's a higher agonist, it's a more potent agonist than DEET. So why could that be? If you look at the binding pocket, again, it's sort of like the game of 10 differences. It's very, very similar, but there are some differences that we can highlight. And the most interesting to me was this methionine in the pocket, which is a very flexible, so very floppy hydrophobic side chain. And we find it in these two structures very clearly in different rotamers. And it's sort of moved up and out of the way in the presence of DEET. And I thought this might give us a bit of a hint because it seems like in the presence of DEET, this methionine needs to sort of move out of the way because otherwise it will clash with the density of DEET, which occupies more of that side of the pocket. So I thought if we mutate this methionine into a smaller also hydrophobic residue, perhaps we increase the agonism of DIT because we make more space for that side of the molecule. So I mutated this methionine into an alanine, and that's exactly what happened. If you look at the DIT curves here, removing this side chain, making it smaller, substantially increased sort of the potency of DIT in the binding pocket, where presumably because now there is a better fit but more space. And interesting, it actually made it worse for eugenol. If you can see here, the shift in the EC50 goes in the other direction. And that's presumably because the removal of the side chains here leaves a space that is now too long for that side of the pocket to interact effectively with the eugenol. So now it's sort of a space too big. So this is really interesting, and it gave us this sort of interesting array, this hardening thought, which is Odor discrimination in binding pockets relies on incredibly tiny details because the rotamer of a single methionine is not what you would call a substantial change. So in order to understand how different ligands activate different receptors, we're going to really need to look at very, very small parts of the binding pocket to get a picture. So just to summarize this first introduction into this family of odor receptors, we have this bristletale OR5 that is a homotetrameric ion channel with a lovely binding pocket that activates in response to 35 different panels. I think it was 35 in this panel, and they're all through the same binding pocket. And then again, if you want to predict odor binding and odor specificity, that's going to be a tall challenge because we're going to really need to see things like the position of the last three carbons in a side chain. And so this is as I said earlier on, this was sort of our work in what is perhaps miscalled the ancestral ORs, sort of the earlier versions of this family of proteins in insects, which are, again, homotetrameric ion channels. But if we want to understand how mosquitoes bite us, we need to look at the ORs in insects that are of more relevance, like mosquitoes. And those are what we would call, more technically, they're neopteran species, or miscalled modern ORs. And something really interesting happened in these receptors. Instead of operating as homotetrameric ion channels, very early on in tree, two of these subunits, or one of the subunits, got fixed into a co-receptor subunit that is called OR co-receptor, and so shortened for ORCO. And this ORCO subunit, it's just as an OR, same family member, doesn't have a binding pocket anymore, or it's not functional anymore, doesn't bind odorants, and there's only one in every insect, and it's the same. So it's a very early event in the evolution of insects that got fixed into this co-receptor subunit that now assembles and is necessary to assemble a proper heteromeric odorant receptor complex. This is what we knew by then, but we have no idea why. What is the role of ORCO? Why did this happen? What does ORCO bring to the equation, and how does it look like? And sort of, again, can we understand something about it? So now in my own lab, we set out to understand the structural underpinning of mosquito olfaction, and for that we need to understand these heteromeric odorant receptors. And by the way, this is how they operate. All odorant receptors operate in things like Drosophila, or just your cockroaches in your house, or all of the insects that you probably run into more frequently that are bristletale, they all have this type of receptors. So we didn't really have structures, we didn't have a lot of biochemistry, but the models for how this work were modeled as something like this. They'd be proposing to have two ORCO subunits, two OR subunits in some sort of configuration, and it wasn't really clear how that happened. So we went on to try to solve this structure, to understand sort of this mechanism. And this is the work of this incredibly, ridiculously brilliant postdoc in my lab, Jiawei Zhao. So Jiawei, we went right, having developed a lot of methods to work with odorant receptors in this family, because we learned a lot from the bristletale ORs. Now we really decided to tackle the mosquito OR, and one of the most relevant ones, because this is the mosquito that transmits dengue, so transmits a lot of other viral diseases, but dengue causes dengue fever, which particularly affects, for example, South America, just why I care about it. It's involved in human host seeking. So this receptor is involved in detecting the sense of humor, the smell of humans, And it detects mostly it's broadly tuned, so it detects a bunch of aromatics. The top ligand is indole. You might recognize indole. It looks a lot like tryptophan. That's because it's a byproduct of a metabolism of tryptophan, which is why it's such an important cue for the animal kingdom. Because if you find indole and you find proteins in degradation, that's organic matter. So that's where a lot of microbes and insects go to. So this is a very important odor receptor for insects. We went straight for it, and Jiawei again brought incredible drive and expertise to this, and right away got an amazing, really amazing trace for purifying the heteromeric complex between OR10 and ORCO, which behaved beautifully at the bench with his expertise, gave us fantastic protein at the grid, really beautiful to the classes, and my favorite slide conversation after many years when you get really happy when you get your structure. Okay, so what did we find with this heteromeric compound? Something really shocking. The heteromeric OR10 orgo complex does not have 2 orgo subunits. It has 3 orgo subunits and 1 OR subunit. This, and for those of you that have looked at densities for a long time, it was very easy to see that because you can see that orgo has these beta loops that are pretty rigid. They show up very clearly in the density. OR doesn't have the loop, so it's very clear to distinguish. It's really unequivocal. You can't miss it. So this was quite shocking for us, and I'd say a lot of the field who went in this array when we proposed this, because it's just very interesting to imagine how a single odor unbinding pocket now is able to gate this channel. So how does this work? Again, remember, ORCO does not have a binding pocket that binds anything. So how does this get open and activated by odorants? All right, so we went on to some functional characterization to solve this. My amazing student, Andy, did some recordings. This is electrophysiology. We record currents in response to ligands. And the first thing you can do, for example, in the ligand series with different ions, you can determine the selectivity, and meaning what ions pass more readily. And the permeability sequence of the ion channel immediately suggested that this passes hydrated ions through, just because of the size of the hydration shell, and it passes cations, and includes calcium. And so that gives us a size of what to expect in terms of the pore. And the other thing that you can do when you do these things in a dose response manner is you can then get in your dose response curve, you can plot and you can calculate your Hill coefficient that, if you remember from your undergrad classes, it is a measurement of cooperativity in a receptor. It's not a measure of number of subunits, but certainly cooperativity. And a Hill coefficient of 1 is compatible with a single binding site in the receptor. So all of this suggests that indeed a single binding site could open this channel, and it could be compatible with the structure that we obtained. So we went ahead and we solved the structure. And what we found, again, really interesting, is that the orco subunits do not move. They're pretty much in the same place. So they don't really participate in the activation. And it's all mediated by the displacement, the movement of the pore subunits in the OR subunit. And that, again, sort of charting the ion conduction pathway, get a pretty wide opening at the entrance that will be compatible with the passage of hydrated ions. So all in all, this could mean that indeed this structure that we obtained from heterologous systems, it could indeed be how it operates in the cells. So very briefly, for those of you who like to look at structures and how they operate, how this is open, and I want to keep, okay. So how does this thing open, or why would odor unbinding in that little pocket lead to anything? So this is a view of the heteromer site and top. The odor unbinding site, if you zoom into this in an incredibly sluggish Mármol movie, you find the binding pocket again in the same place as in the OR5. It's again, if you look at the residues that are directly interacting with the ligand, they're mostly hydrophobic residues. It's a very small binding pocket again, and it's mediating, as it was before. There are some aromatic interactions, some pi-pi interactions, and in this case, just one single hydrogen bond between the hydroxyl of this ligand. This ligand is ocresol, it's not indole. We had other ligands. Again, it's a broadly tuned receptor, so you can solve structures with many different ligands that are similar. And so again, the binding of these molecules is mediated by hydrophobic interactions. And then if you try to figure out how exactly binding, in this case ocresol, stabilizes the open conformation of the channel, you have to look with kind of like a a glass because it's a very small movement, but I try to highlight it here. You can see that if you look at the subunit, at the OR subunit, most of the protein here, most of the protein, the distal protein to the pore remains the same. And the three helices, which look kind of a mess here, but I'll walk you through it. The three helices that connect the binding pocket with the pore helix are the only ones experiencing some sort of movement. So what's happening here in the unbound state, which is in red here, and in the bound state, which is in yellow, you can see that there is sort of a collapse of the pore helices onto the binding pocket in the bound state. And what happens is that the ligand binding in the pocket, it stabilizes a more contracted conformation of the pore that leads, that doesn't sequentially lead, but it stabilizes a more compact conformation. And that's due to the fact that these three helices here connecting the binding pocket and the pore there are three completely coupled helices. They're nearly glued together. So this is the movement that I was referring to. And these helices are glued together by hydrophobic interactions. Here I'm showing all of the residues in these helices, and you can see there's only hydrophilic residues lining the pore in this helix here. Everything else is sort of like a hydrophobic glue. So these three coupled helices, if the binding pocket is stabilized in a contracted conformation, the whole thing ends up opening the pore by displacing the pore helix. So lastly, about this sort of heteromeric assembly, I find it really interesting that the ORCO subunits are not doing anything, meaning they're not interacting in any way with the odorant or moving in the pore, or at least in these structures that we obtain, we don't really see any movement. And we really, we began with this question. What I'm showing here, for those of you who might not be clear, this is the RMSD. So this is is highlighting the regions with higher RMSD between the apo and the bound state. And you can see that nearly all of the movement occurs in these three helices that I mentioned in the OR. ORCO subunits in apo and bound states remain nearly static. So what are these things doing? If we look at a sequence alignment, this is a bunch of ORs from all different insects. And you can see that the majority of the conservation is constant, happens in the C term of the protein. If you map that onto the protein, that is the only region where the OR interacts with ORCO. That's what we call the tetramerization domains and the pore. All of the diversified region is the rest, sort of the odorant binding site. So one thing that we thought that perhaps explains why ORCO is there is that the existence of ORCO provides sort of like a stable scaffold onto which you can then diversify your OR, whose only job is to navigate the ion conduction pathway. So in a way, if you just keep the interactions pretty conserved across OR, then you can diversify the rest of the protein in order to accommodate the billions of odorants in the space and still be able to operate a tetrameric ion channel, which is kind of a tall challenge. So this is a hypothesis on sort of why the emergency of ORCO subunits might have been advantageous. So it's a small summary of this part. These heteromers assemble as 1 and 3, 1 OR, 3 ORCOs, at least in these expression systems. By now, I should have put here more citations, but we got this structure, and then there's been 3 other groups that got subsequent structures, and also we did unpublished ones, and all of these are 3 and 1. So everything we can make in the lab is 3 and 1. We do think that there are other stoichiometries possible, and we have some evidence, and other labs do as well. And so it'd be really interesting to understand why would you have 3, 1, 2, 2, what does it do, how is that different? We'd love to see those, so we're working on that as well. And then why ORCO showed up? A possibility is that the fixing 3 subunits without allowing diversification just keeps them stable and allows the OR subunit to just simply operate the gate in response to odorant binding. That's our working model for this, and I think it's a pretty interesting topic. And so the last 10 minutes or less, I just wanted to briefly touch on some other modes of odorant specificity. I've talked a lot about OR5 and OR10, which are broadly tuned receptors or promiscuous. They bind many different types of molecules. But I mentioned that there are some other receptors that are incredibly important for ecological reasons that are selective receptors. These are typically receptors of things that are toxic, for example. So you really need something that detects that right away and sort of bypass. So it's an innate response, for example, to toxic compounds. So we wanted to understand how this happens. How can you get this binding pocket to be only responding to one type of compound? So for this, we focused on, and this is the work of another amazing postdoc and an amazing technician in the lab, Steven Sanjana, that they focus on the classical, the most canonical selective receptors in the animal kingdom. These are pheromone receptors. Pheromones are compounds emitted by individuals that mediate courtship and mating behaviors. They're volatile, and so the signal, I'm showing here the moth because it's actually moths is where these compounds were first described. I found it interesting that pheromone sort of has this etymology, which is sort of like transferring your excitement to somebody. So that's a pheromone. It's a volatile compound that transfers your excitement, sort of like a nature's perfume. So how does this happen? All right, so here's the moth. This is the canonical pheromone. This is Bombyx mori, the silk moth. This moth has to recognize these two compounds, bombicol and bombical. It's the mixture of these is what it responds to. It needs to be, as you can tell, bombical and bombical are very fatty compounds. Their hydrophobic tail is exactly the same, and they differ only at the tail end, where you have either a hydroxyl or an aldehyde at the end. Okay, so we had the oxidized form. It's just the difference is the oxidation of the last group. How can this moth distinguish these two compounds? Well, in the olfactory sensory neurons, it has in the sensilla that houses the olfactory sensory neurons, there is two neurons, and they have two receptors, OR1 and OR3, that have entirely opposing selectivities. One is very specific to the alcohol, the bombicol, and 3 is very specific to the aldehyde. And they have, each one of these receptors has a lower potency or is less activated by the other compound. So essentially, we have a pair of receptors that has opposing selectivity for the aldehyde and the alcohol form. So this has been known for 40 years. And in fact, you can make these mixtures, and that's how you maintain your moths when you want to make silk, I guess. And so how can you get a receptor that recognizes whether this has a hydroxyl or an aldehyde? So I focus on this one, the aldehyde. From a chemical perspective, it's more interesting to me because if you think about, an alcohol has many more hydrogen bond possibilities than an aldehyde. So how do you get something that's more sensitive to an aldehyde than an alcohol? You would get more sort of enthalpy of binding with an alcohol than an aldehyde. So how do we get this? We solve the structure. Again, has 3 orgos and 1 OR, just like any other receptor in this family. And it looks just about the same as everything else I've shown you so far. So we're going to look at the binding pocket and see what's the chemistry here. Here is what the ligand looks like in the binding pocket. First sanity check is a beautiful, longer, larger binding pocket that really accommodates the whole shape, including the unsaturations and the geometry of it. All right, so that's great. So what's happening at the business end of this compound? So here I'm showing the density for the ligand, and it's right next to this lysine here. So if you dock bombical into the density, it doesn't quite go very well. It sort of gets out of the way a little bit and doesn't fit the density very well. If you dock the alcohol in the same binding pocket, it shows you some nice hydrogen bond interactions with the threonine and the lysine. But so something that the lysine can do, because those of you with some memory on the chemistry of this, an aldehyde can form a Schiff base with a primary amine like those present in lysines. And that modeling of a covalent bond between these two forms a beautiful density from a beautiful adduct that is fitting the density quite well. So we're thinking, all right, so this is a Schiff base. So the aldehyde establishes a Schiff base with the lysine. Can we prove that? All right, so if we take sort of a mutational approach, if you mutate the lysine into alanine, it kills activity for both the alcohol and the aldehyde. Makes sense. It seems to be involved in binding of these two compounds in any way. But now if you switch your alanine you mutate the lysine into a glutamine, you completely restore activity for the alcohol, which is predicted to interact only through hydrogen bonds that are perfectly possible with a glutamine, but it does not recover activity with the aldehyde, meaning that the primary amine of the lysine is fully necessary. It's not just a hydrogen bond partner. It's a covalent adduct with that. Now, the other thing, so we have this covalent bonding. But now if we looked at the rest of the chemical environment around the binding pocket, we see something else. There is a cysteine right behind the imine carbon here. Could this cysteine be involved in some way? In the modeling of the pocket, the cysteine could be stabilizing the imine bond through a hydrogen bond. Another possibility, because cysteine is also a nucleophilic, has a nucleophile head, the cysteine could be attacking the imine carbon to form what you would call as a thiohemiaminal. What's happening here? If you mutate the cysteine into alanine, you lose some of the activity, meaning that the cysteine does have a role in this interaction. But if you mutate it into a serine that is actually a better hydrogen bond forming partner, and it has the same distance, you don't recover activity, meaning that the cysteine is not involved in a hydrogen bond formation. It's actually forming another covalent adduct. And if you model that into density, it's a perfect fit. So what we're actually doing here is modeling exactly what we see in the density. We see a covalent adduct between the lysine, the cysteine, and the aldehyde. This is not strange. So this is a potential mechanism. We're now probing chemical mechanisms, so I don't want to go and tell you that this is what happens. One potential mechanism is a subsequent sort of nucleophilic attacks by the lysine and the cysteine that will give you this last adduct. But what I want to highlight that's interesting to me, and this is how the density will look like, is that this also happens in many other systems. You are probably familiar with rhodopsin and the retinal reaction that occurs. That's how we perceive light. And in this case, if you look at the binding pocket, retinal is an aldehyde that is bound to the lysine through a covalent bound to a shift base. And that shift base is critically stabilized by the glutamate residue that is charged and that is pairing with the positive charge of the imine bond. If you mutate a glutamate in rhodopsin, you lose activity because retinal bond is too unstable, and so it gets hydrolyzed right away, or it's a very rapid reaction. So you need to stabilize the shift base through a glutamine in this case. Otherwise, you don't have a stable enough interaction. So what we are here seeing is that we have the same situation where we have a shift base formation between the aldehyde and the lysine, which is only possible with the aldehyde, not with the alcohol, and is in this case stabilized not by a positive charge, but by a cysteine that is involved in the second adduct formation. And then lastly, is this perhaps a more universal mechanism of aldehyde recognition in the olfactory system? Well, now we have AlphaFold, so we can look at binding pockets of a lot of other things. And if you look at the OR17 receptor in mice, this is being classically described as detecting octanol, an aldehyde. You can, dog or predict. In this case, I think we used Chide and we folded it with a ligand. The binding pocket has a lysine in it, right next to the aldehyde in the predict position, and it has another charged residue that is possibly also stabilizing this adduct. So again, this is a prediction. This is not a structure, but what we're saying is that the binding pocket of the aldehyde has, again, a lysine and an important charge stabilizing this adduct. So, all right, now the final summary of everything here. We have seen a lot of promiscuous pockets for hydrophobic compounds, and these rely on flexible hydrophobic interactions with some aromatic, some pi interactions, but generally a very promiscuous specificity. We have some sort of very selective binding pockets. In this case, the one for aldehydes that seem to rely on chemistry, because nature loves chemistry, and so do we. And this is how you get a very selective pocket for an aldehyde, something that alcohols could never do. And then lastly, the whole family of other receptors that we started to understand as a homomeric family and now into heteromers, that is really giving us a lot of thoughts, a lot to think about on how sort of evolution of these proteins can better serve the diversification of the binding pockets in order to allow for the detection of a large chemical space. So with that, I'd like to thank just so many people. The lab is fabulous. You all, work in science. We have the best job in the world, right? Even in these times. This is an amazing group of people. They're incredibly talented. I've highlighted names of the people directly involved in the work I've shown here, and then the funding sources, and all of you for your attention and invitation. Thank you.
Thank you, Josefina. That was a great talk. Let's start the Q&A. Jason Kalber had a question. Can you bring us a question up from the chat? Jason runs the cryo-EM facility in the basement. Does the change in the consensus structure, eugenol DEET versus APO explain the conductance, or is the change in dynamics also important to explain conductance change?
So if the change in the binding pocket explains the change in conductance of the channel, yeah, that's really interesting. No, I mean, we don't understand why some, so we see the same open structure. We don't have differential open structures with different ligands. So, and the binding pocket, we can see more or less interactions, but we don't have any idea of why. And we don't really measure affinities because we don't make enough protein to do ITC or any more quantitative techniques. So we don't really know, we can't, we really don't know the actual affinity of these compounds and whether that is directly related to the differential conductance, but the pore looks exactly the same in both open states. So the mechanism is the same, it's just different efficiency for both ligands.
Does the ion selectivity change at all?
No.
It's
No.
always the same.
The ion selectivity in this channel seems to be determined by simply the size of the pore. There is no selectivity filter because it's a non-selective channel and it just passes ions based on the size of the hydration shell. And both the usual and the DEET open structures have the same ion selectivity.
Right. The binding pocket, the cavity, is that's probably doesn't accommodate water in the absence of a ligand.
What's in it, right? What's in the binding pocket in the absence of a ligand? Yeah. Yes. So the APO, in OR5, the APO pocket, we don't see any density. So whatever is there is to move. It's not enough to give us a solid density. It might be really poorly accommodated water, but I don't think so. In the OR10 pocket, that's a much better structure. In the APO state, we actually see the head of a lipid that comes through the membrane because the binding pocket opens into the membrane.
Okay.
And we see a lipid.
So these hydrophobic ligands are entering through the lipid bilayer.
They partition into the membrane. We think they partition into the membrane, and we see the direct access, which is why we see this lipid head into it.
Right. Sang-hook.
All right, thank you so much. I have a follow-up question to Jason's. So I'm wondering about difference in conductance dependent on the stoichiometry of the because for ancestor OR specifically speaking, right? So even though you showed like cryo-EM structure with everything, all the binding pocket were occupied by the ligand, but I think in physiological condition, you might consider, right? You can think about all different stoichiometry, right?
Mm-hmm.
So I don't know whether you can actually differentiate those different conductance from your patch clamp experiment or the electrophysiology experiment.
Yeah, that's an interesting idea because in a way, if we're saying that the selectivity of the channel is directly related to the size, so it's just a diameter thing, the different pore helices and how much they open will give you different selectivity. That is certainly true for different ORs. So if you make a tetramer with different types of ORs, you get different selectivity, meaning that the identity of the helix of that OR would affect the selectivity. But we've never really thought of trying to measure selectivity for different stoichiometries. One of the first problems is that we don't seem to be able to make any other stoichiometries. So in all of our structures, most of the particles are 3 and 1, and a very small percentage of the particles we can resolve beyond 8 angstroms, but it looks to be 2 and 2 just because of the lack of loops. So we don't have enough population of different stoichiometries in order to be able to detect them functionally. But I would be really interested to stabilize that and see if we can measure, and that will give rise to different functional states that perhaps will have different functions in the neuron, and that would explain what different stoichiometries might exist as well.
Then might dictate then the binding is very cooperative, therefore coefficient
Yes.
might be very different from one.
Yes. Absolutely, yes.
Could you repeat what Sang-hook just said, please?
It's a very different Hill coefficient for this binding event of 2 ligands versus 1 ligand, for example. Yeah.
Hi, I'm a systems neuroscientist. So I'm going to ask the systems neuroscience question. I couldn't help but start thinking about that beautiful graph that you showed at the very beginning about like the TMCs and the piezos and the beautiful analogy to frequency and waves, right? And then I started wondering about like, wow, we have so many that have to do with odor and taste. And so I'm just wondering about evolutionary pressure,
Mhm.
because of course then you then shared all of these dendrograms, and so I started thinking about that. So evolutionary pressure at the protein level versus at the circuit, right? So valence is important, right? So you alluded to the smell of coffee. Some of us like it, some of us don't. So valence is, I think, a systems neuroscience question. But what's the evolutionary pressure here? And then how, and then can you elaborate on that for someone like me that doesn't think about this, when that extra subunit arose in time, and then you see kind of this explosion
Yes.
of organisms that use this.
Yes.
So
Yes.
Mhm.
Yeah, it's a really interesting question. And I think I like to think about this a lot. So one thing, I'll tell you some things we know, because a lot of it's speculation, but the species that have no orco at the root of the tree, the sequence diversification between ORs is about 40%. That's actually very high. The sequence, I mean, not diversification, the sequence conservation, so the conservation numbers of ORs in things like mosquitoes is down to 10%. And so one of the things that we think about in terms of the evolutionary pressure is that if you are going to have no fixed subunit, your ability to diversify, to make sort of the sequence much more diverse, drops, because you're messing up with the functional aspects of the channel and you just kind of lose functionality. So once Orco showed up, the diversification exploded in the insect tree. All of the species that have Orco have many more ORs. And so we think that that really enables the diversification. And so you don't have to rely on diversification so much at the circuit level, and you can do a lot with the proteins. This is a very rapidly evolving family of proteins. That's known as sort of the birth and death model of evolution, where there is new ORs coming all the time and ORs dying, and the neurons are more or less always the same. And the modulation of valence occurs downstream, most of the time not at the periphery.
So then do you think that then that diversification allowed valence to be encoded in the periphery, and for whatever reason that increases fitness?
Yeah.
Why? That makes no sense to me. But anyways, I'm a neuroscientist. So.
I would love to answer that, but one thing about the bristle tail that was a problem that I had is that we don't really know if it's repelled by DIT, for example.
Right.
So we don't know what is this receptor binding eugenol and DIT. We don't know if it's repelled by DIT, and we don't know if another receptor also detects DIT in a different sort of inhibits, and maybe the combined response gives you something different than eugenol. That's why I sort of moved away from bristle tails, because they're beautifully biochemical, but we don't have. I try to get bristle tails. You gotta go catch them in Switzerland. They don't grow in the lab, so I decided not to go that route. So I don't have any idea of the valence of eugenol and DIT for the bristle tail, but it would be a great place to study it, because we're gonna have 5 ORs, so we could do a lot more.
Thank you. So Jason Kilber wants another bite at the apple, and he asks, he states, I want to try asking my question in a different way. When eugenol opens the pore, 2 things might happen. There is a shift in the diameter, aperture, and the channel could become more flexible. Does the pore wall become more flexible, or is it just as rigid as in the apo state? Is the diameter of the hole big enough to explain the ions that get through, or does molecular breathing also play a role?
Mm-hmm. Yeah. The pore, I didn't show the whole pathway of the pore. The entire pore is a lot larger than the gate. The gate is very small. It's the last 3 residues of the pore helix. So the rest of it is just widens right away, and it's a water-filled cavity throughout. So the gate, which is also the filter, is at the top, and the very small movement of just one, or in the homotetramers, 4 of them, and even with some baseline activity. So in the absence of eugenol, you have some openings, and that's because, again, it's such a small movement that you don't have such an energy threshold. So without ligand, you have a baseline activity, which is a known feature of odorant neurons. They have sort of a baseline spiking in the absence of odorant. So the pore is breathing, is flexible, and it breathes. And when you add eugenol or any agonist, you stabilize sort of the helix out thing, but not by a lot. It's a very low threshold movement. I don't know if that's so you're asking if it's either the dilation of the diameter or breathing, and it's both. At least that's what we can see.
I have a from a molecular biologist. Hello. So kind of thinking about this in a competition way, right? So you're only providing the best substrate and then seeing it binding in the pocket. But what if you provided it with like the top 10 contenders? There has to be something about it, right?
Yeah.
That because in the real world, you're exposed to all of these at the same time. And so would you ever see kind of competition for binding within your cryo-EM?
Yeah, I don't want to do that. The reason I didn't do it, and I don't think I want to do that, is because a lot of my issues at the beginning of this work is how am I going to distinguish the density of a usual and a DEET? Because they're not, I don't even think there is a problem. The limitation is not technical. I could pick 10 million particles of these, and I would still see the density pretty flexible because this is theoretically not tethered. The ligand is kind of floppy inside. So usual and DEET really differ by 3 carbons. If I put them together in the sample and I look at the binding pocket, I'm not sure I would be able to distinguish, and I probably will get a timed average of some bunch of them. And so I want to know that because that's how it happens in the real world. There is a mixture of compounds, and possible that's part of how they work. But structurally, I don't know if that's how I can get at it. I don't know how, but yes, I agree.
I'm going to take the chairman's prerogative and ask the last question, please. Is there more than one orco?
No,
There's only one.
There's only one. In all species, they're interchangeable. You can take a mosquito orco, put it in a wasp, and no difference. They're about 80% conserved. It seems to have appeared very early on in the tree.
And is the sequence identity high then among all the orcos?
Yeah, it's 80% conserved.
80% across all the orcos.
Yeah, in all the insects. The other thing that I didn't make the system, right? There is only one gene, and if you mutate a gene, the animal doesn't smell. You think, you could get a spare one, but no, this is one. It's just one in all insects.
All right, fascinating. Thank you very much. That's great
Thank you
talk.
so
Let's thank
much.
Josefina again.
Thank you.